So there does seem to have been maybe a mild level of confusion about when the test was due. That I'd said something that it was due on Monday, and then the thing on Canvas said that it was due last night. So if you haven't, although most people had turned it in, but I reopened it for for tonight. If you'd like to pull something back out and revisit a question or two because you felt like you were a little bit rushed, then that's okay. Knock yourself out. If you have a hard time with all of that, then just email it back to me. That's fine. So I don't really care. Brady's just like, whatever. Oh, it's a test. Just do it whenever you get a chance. It's fine. Like, nobody worry about things. So
no, I accidentally, like, my laptop died and I was 80% done.
Oh, yeah.
And then I get back on to finish it last night and there was nothing there, so I had to restart. So I was in a rush.
oh no, Brady.
I was really upset.
Yeah, I got text in the group chat.
I like
Have y'all let the biomedical engineers in on the group chat?
I don't even know whose number it is anyway.
yeah, I don't know who anybody is.
Jordan, Josiah, y'all in the group? There's apparently a group chat, which I immediately became anxious about. Why are y'all all group chatting about the class? But
It's
maybe you all should be added into the chat. So
It's like mostly
it's the
for stats.
okay.
Because I think I'm the only one that isn't in stats that's included in
I think so. I don't know. It's a random thing.
Okay.
Yeah.
Logic should be.
You went to the mock that day, you got
it. You want us?
It's not that, it's not.
No.
Okay.
I'll get it.
You guys can get added a little bit later on. Okay, so we've been talking about resistance training, all right? Specifically in resistance training, we've been talking about neural adaptation. So how the nervous system changes, how it communicates with muscles. I ran you guys through some of the kind of more important and kind of historical research for some of the first ideas on what the things that change are, kind of how we began to notice. So I thought it might be useful to just make a summary. Can you guys envision now a test question where I'm like, hey, talk about things that change and talk about how that would make them go up. Here's your list, okay? So we can increase motor unit recruitment, right? So more muscle can get turned on. Maybe we can drive it at a higher firing rate. There's a little bit of evidence for that, okay? So we see specific adaptations where we get better. If we train at a fast velocity, we get better at contracting at fast velocities. If we train slower, we get better at contracting at slower velocities. There's a little bit of cross with those, but that is also going to be very specific. We seem to shut down antagonist co-activation as part of things. So if you train your quads, you learn, quote unquote, to turn off your hamstring. So more of the force from the agonist muscles get applied to the external load. And a lot of this potentially seems to be driven by increased descending drive from the motor cortex and an increase in excitability, or it's easier to turn on the motor neurons in the spinal cord. Okay? So I get a bigger signal from the motor cortex. That may be helpful to get more to turn on. And then also it becomes easier with any amount of signal to get those motor neurons to go. Okay? So that's kind of the summary of the neural adaptations. I don't have on here, I know we mentioned it last week, there is some evidence that Golgi tendon organs get downregulated a little bit in the midst of this. That's also probably what's driving this decreased antagonist activation. Okay? So huge amounts of research going into all of these kinds of things. If you want more specifics, I'm more than happy to try and get some of that stuff. Okay? Or you can take Dr. Pereira's neuromuscular phys PC level class in spring. Okay? I don't know exactly how much he covers, but he will cover more of all of that. And may also do some practical things if you guys want to practice and like see some of the techniques and stuff that we have mentioned. Okay. Okay, so that's the nervous system side. That occurs in, within a couple of bouts of exercise. It accounts for most of the strength increase that we see in the first, say, 4 to 6 to maybe 8 weeks of a training program before we see actual gains in muscle size. Okay? So let's move away from the nervous system. Let's talk about muscle. Let's talk about the thing that all the bros that are over there today, right, doing their bench press, what is the thing that they're hoping to gain most of the time? And that is this contractile protein adaptation, right? This is what we call hypertrophy. This is making the muscles bigger, okay? Making them bigger. I learned this morning in my undergraduate class that there is apparently some sort of oil that people will inject into their muscles to make them bigger. Do you know what this is?
Yeah, more than their muscles too. It's like big in like Russia, I've seen a lot.
And what is it called? I still have it in my phone because we had to Google what it was, but it's like this oil, and you inject it in there, and it never gets cleared.
It's called synthol.
Yes, synthol. Thank you, Ethan.
Thin ball.
Synthol. Synthol.
Why?
How the hell?
Hello.
Float better?
I don't know if you float better, but it like gets in your muscles and then it hardens. And you then like can't make your muscles bigger on their own, and it like can damage the muscles. You don't get any stronger. You just get big and swole. Christmas. Did you Google something with it, Brady?
No.
So okay.
It's terrible. It just eats away your muscles and causes
Okay. Do you know someone that has had this done, Ethan? Or you just seen pictures or things?
Whoever said Russia, right?
Yeah.
Russia. The guy from India is like,
Okay.
just lives off of that.
Okay. The guy this morning that they showed me a picture of, I think was Brazilian or something.
Wait, can I see?
Just some big muscly dude with a beard whose biceps were like, I don't really understand.
The heck?
You can tell that.
It just looks straight up it.
And
Yeah.
after a couple of years, it's
Like it doesn't go away, like it just hardens, like you're just kind of stuck. Yeah, weird stuff. So what those people are trying to get is this, okay? They want more actin and myosin, and they can't get enough, so probably they just inject the stuff in there. They want more actin and myosin. The key thing is that we're going to get an increase in cross sections. They get the fibers themselves get wider, the diameter gets bigger, okay? And that gives us more fibers and more cross bridges in cross section, which should theoretically then give us more force. Brady, did you find something ridiculous?
It just says synthol kid mixes in there to say you. It's just like, I don't know how to describe what it looks like. It looks like a tumor.
And in some ways, it's kind of what it is. It's like a big tumor. Okay.
They do have like ones that are terrible. You see if you buy it over.
Uh-huh.
You just got tattoos to make it look like he's defined, but it's all it is.
Interesting. Okay. You know, so the thing about cross-sectional area increases, is I'll show you guys in a couple of slides. The reason people use the synthol and these synthetic things or steroids is there's a limit to how big we can get. Okay. And it takes a little while for those things to occur. So generally, we're probably going to take maybe 6 to 8 weeks to see measurable increases. One of the few studies that's used high resolution imaging was done in our department. It was done, I don't know, maybe 10 years ago or so. And they did some bicep stuff, and they used Dr. Deb's peripheral CT machine. So you put your arm in the CT machine. And I think at about like 4 weeks or 5 weeks, they began to see something that they can measure on the CT machine. It's very, very small on that. So for most people, like if you want to see like I gain weight, or I can put a tape measure around my arm or my leg or something, probably looking more in the 6 to 8 week range to see this noticeable increase. Okay. As I said, there is a limit. We don't have a lot of data on what that limit is, but we assume there is a limit. Otherwise, it would all look like synthol dudes, right? You just go and you lift and you just get as big as you want. That may also be the thing I think that drives a lot of issues with bodybuilding or kind of layperson ideas about this. Is they just think like, I can just go lift and I can get as big as I want, which is just all bullshit. There's no way, right? Most people are going to be somewhere in the maybe 5 to 10 up to maybe 40% range if you're looking at kind of one standard deviation around the mean on things with all of that. I'll show you all a little bit of data there. We think a lot of it is genetically driven, okay? A lot of it is genetics. And so diet plays some level of a role. What you're doing from a resistance training standpoint plays some role, but a lot of it is if you've got good genes, and we know what some of those genes are, okay? If you've got good genes, you don't make much myostatin, right? You have some high activation in some of the mTOR pathway genes and things, then you're going to be a big responder. Other people, just not going to be a big responder. Doesn't really matter, okay? No matter what it is that you're going to do. And one of the issues that we have a hard time understanding the genetic limit in those things is steroids, okay? I don't know that I have the data in there. If I don't, I'll try to pull it for you guys and have it for next week. There have been a couple of studies where they have done randomized control trials where they've given people steroids. It's a very famous one. I believe it was by the first author's last name was like Basmagian or something like that. But it's in the New England Journal of Medicine, which is like the best journal in the world. But I mean, they gave people steroids and had a group that sat on the couch and didn't do anything, had a group that did resistance training, and then there's a group that got a placebo, I think, in all of this. And so we know some level of what the responses are. I can't believe an IRB level do it. We could never do that. We could never do that, right? Can you imagine how many bros we have who are like, dude, I want to get those steroids, right? If they want, like, we're going to give you guys Winstrol or some kind of other horse steroids. I was like, you know what Winstrol is? So it's just Google Ben Johnson in the '88 Olympics. He was on the Winstrol. It's a horse steroid. I mean, in some ways they're all horse steroids, but so we have a little bit of data. But we don't have a lot because it's just unethical for us to study these things. And it's hard if we want to try to do like case studies or we're going to look at people that have a lot of muscle mass. A lot of them have taken steroids. So it's hard for us to separate what can you reasonably get naturally, quote unquote, versus what may be the effects of steroids. So we have a little bit of data. So we're not going to focus too much on kind of the upside, but we will talk a little bit about what do we know about sex and reps and weights. What are some of the molecular signaling pathways that we know are involved in a lot of all of this as well. Okay. These are again more data from the Monica Hubel and Priscilla Clarkson study. If we want to look at things that I showed you guys when they talked about 1 rep max NBC games. This is in the bicep. This is MRI. There's our nice normal distribution. Here's CSA change, right? We've got men in black and women in the white, I believe. So we're going to have an average of somewhere between about 15 and 20% of an increase in the midst of all of this. Okay. So I know we talked about that earlier in class, but if this is the average, an average person can gain 15 to 20%. You increase every muscle in your body that big, and you're going to put on like 8 pounds or 10 pounds or something. Okay. So most people cannot get like as giant as what they think. They're not going to look like our real bodybuilders. There are some people that are way out here. Okay. 50, 55% increases in size. You've got hyper responders. There are people that are losing mass. I don't know what the hell they're doing. But what we're going to try to talk about in the next kind of things, though, is kind of what gets into and controls what this process is going to do. So, in order to appreciate, this may be more information than you guys really want. I think in order to appreciate what happens, okay, the things that drive hypertrophy, we first have to understand how we might go about quantifying both hypertrophy and the different models of trying to study the things that make muscles get bigger. So we're going to talk a little bit about how we measure hypertrophy. And then there are some in vitro cellular models. There's some other kind of lower animal models of hypertrophy. And then there's people. And so hopefully what you guys can appreciate from this is this whole thing is a mess because it's hard to have really good experimental control. And so that limits a little bit about what it is. Okay. So there are some invasive ways that we can try to quantify muscle hypertrophy. Primarily, we can take a muscle biopsy, okay? You guys might be willing to give me a muscle biopsy. you're probably not going to be willing to give me an entire muscle, right? Because if I want to just take all of your biceps brachii, are you going to be cool with that? We just cut it all out? No? Okay. Not a great way to do things. So one of the things that we can do is we can look at myofibular cross-sectional areas. You can look at a single muscle fiber. I can get that from a biopsy, or I can take out a whole muscle and then tease out one individual fiber. Okay. There are some limitations to this thing. Is what happens in one cross-section or 20 cross-sections from one small area in your muscle, indicative of that entire muscle, right? We take a little chunk of size quad from his vastus lateralis, right? And there's maybe 100 muscle fibers in it. And we put them under a microscope and we circle all of them and we make Brady count all of them and measure everything out, right? It takes a while. But the question then is that one small chunk of those few fibers out of the hundreds of thousands that are in there, are they representative of the entire muscle? What if we just happen to get some that didn't get bigger? What if we just happen to get the ones that did get bigger? So there's some issues surrounding all of that. Okay? We can look at myofibular protein content or concentration. I could take your biopsy, I could grind it up, and I can measure how much actin and myosin is in it. I can weigh it. Okay? Doesn't work that great for a biopsy because it's not the whole freaking muscle. If I have the whole muscle, I can grind up the whole muscle, I can weigh it, I can figure out how much is in there, I can get total protein content or myofibular protein content or concentration. I can do this in lower animals. I can have, they're genetically identical. I can have one, it trains, it does something. I can have another one that doesn't. And I can feel pretty confident that I can get a good answer. Again, this doesn't tell us a whole lot in us, okay? In us. And as you guys maybe will see, there's lots of things that result in hypertrophy in single or isolated muscle cells. There are things that result in hypertrophy in rats and mice. Don't work in us. Okay? Don't work in us. And so it makes the study of all of this a little bit tricky. Okay? And the big thing here, right, is are they representative? Is the animal model or the cellular model representative? And is what we're seeing actually representative of the whole? So that leaves us that if we want to study this in people, we need a relatively non-invasive way of doing things. Okay? Relatively non-invasive. So can I take the volume of the whole muscle, right, with CT or MRI or something like that? They make water displacement. You guys ever seen this? Like you dunk your arm or your foot in a water thing. I went from, during my dissertation, quantifying the volume of quadriceps muscles using MRI to doing my postdoc where we were putting people's arm in a bucket of freaking water, okay, to quantify how much swelling we were getting out of their arm. I felt ridiculous every day of my postdoc doing that. I'm like, this is nuts. But you know what? We got really good at doing it, and we found some differences, right? So the whole muscle volume is going to be relatively indicative, much more indicative of physiological cross-sectional area from the back than an individual cross-sectional area. Because what if you don't get equal hypertrophy across the length of the muscle? What if we just happen to image you in the one place where you get really big or you don't get big at all? Okay? So having more is better, but these things, there's a little bit of this that's in there. This is also expensive though. If you want CT, you want MRI, even if you want to do a DEXA scan and try to circle the limb volume for the DEXA, those things are pricey. Or you can just measure girth. You can measure a circumference with a tape measure. We have some relatively inexpensive-ish ultrasound machines downstairs. You can put the ultrasound machine on. You can measure muscle thickness. Those things work pretty well too. You can make multiple muscle thicknesses, and there's some equations. You can try to estimate a volume from those kinds of things. There's some stuff that we can do. Okay, but the problem that we have is none of these things are perfect, especially in people. None of them are perfect. There's a lot of other things that can go into all of this. Okay, Gabe decides, you know what? I'm gonna drink a lot of water today. So he gets more water in his muscles, and then we're gonna go freaking image. What the hell happens, right? Now he's swollen. Or the problem of the study where they found that they did here, like, oh, we're seeing increases at a month. they were doing Con-Ec exercises. They measured them like the day after they've just done stuff. What if they had muscle damage and they had swelling because of all of that? That's what they were measuring. They didn't have a control to know the difference on all of those kinds of things. So it's tricky to do this and do it really, really well. And so that limits a little bit of what we know. I just want you guys to understand all of that. Okay. So much like VO2 max, the things that drive or control the size of skeletal muscle are integrated. There's multiple signals and multiple things that are going on and interacting together that determine all of that. And so because of that, it is a bitch to study, because how am I going to get you guys to all have the same diet, to all walk the same amount, to all have the same metabolic rate? It's almost impossible to accurately study and control everything. Okay? You tell somebody you're in this randomized control trial, you don't know what you're doing, and then what if they work out harder as a part of this than they would normally, right? So it's hard for us to control all of these things. Okay? We think these are potentially some of the factors that there's evidence that supports. We know that the peak muscular force during contractions is important. Okay? How important? I don't know. It's hard to say, but we know it's important. We think there's evidence that the total volume, so of work that you're doing, the number of repetitions multiplied by the force. Okay? So are you lifting a heavy weight? That may matter, yes, in some instances. Are you lifting a heavy weight as many times as you can? That may also seem to matter. There is some evidence in some models that contraction type matters. Looking at people that are doing concentric only exercise, Con-Ec exercise, or eccentric only exercise, there's a little bit of evidence that that matters. It's not super conclusive, but it's out there. There's some evidence that the metabolic demand matters. That actually going to fatigue, going to failure is potentially one of the metabolic signals that interacts in that mTOR pathway that may trigger hypertrophy. There's also some evidence that it doesn't matter. It's tricky. It's hard for us to know. This last one, we really get ourselves in trouble. Side, cover your ears. It's possible that the local and systemic hormonal milieu matter. Ready? Are you in it though? Okay. Dr. Deb has a very particular thought on all of this. My reading of the evidence, not being an endo expert, is there is some evidence that may refute her thoughts on all of those things. And there is some debate. Does the amount of testosterone, growth hormone, IGF-1, and things that get released during and after a bout of resistance training, do those things matter? Maybe. Maybe they matter. They certainly matter if you don't have enough. They certainly matter if you supplement them supraphysiologically. Okay? But Cy goes and he goes and does squats today, and he releases X amount of testosterone and growth hormone and IGF-1. Is whatever happens during that one bout, is that going to drive how much hypertrophy he gets? It's difficult to say. Okay? It's difficult to say. Because we can do and work out in different ways and alter this. And sometimes that seems to matter, and other times it doesn't seem to matter. Okay? So there's a guy at McMaster, named Stuart Phillips. Lots of sort of one of the resistance training gurus. They have a lot of evidence that this doesn't matter. But there's some other evidence that says it does. So we'll try to kind of work our way through some of that. I know that Dr. Deb thinks that in some instances it does matter, and she thinks a lot of stuff that Stu has done has some flaws in it, and it very well may. Right? So we just want to be aware of these things. The problem that we have is that most studies attempt to isolate and control one of these factors. I'm going to hold everything else constant, and I'm going to manipulate peak force. Okay? It's a reductionist model of doing research. Everything else constant, I'm going to manipulate metabolic demand. Okay? All of these things can be done. The problem is the bros over there aren't working out like that, right? Ethan has to train his athletes. That's not what they're doing, right? And so it's an integrated process, and it's hard for us to try to understand sort of what is the most ecologically valid, what's the model of studying this that best replicates real life situations for clinical populations, for athletic populations, for older populations. Okay? It's nice for us to know this signal matters, that signal matters, and maybe we can manipulate it by doing this or that or whatever. But if I can't actually do that in practice, then does it even really matter, right? So we just need to keep these things in mind. Okay. All right. I assume that all of you remember some level of intro biology, and you have seen the idea of transcription of messenger RNA and then translation into proteins. You are aware of these cellular processes by which we make new protein. Yes? Okay. We have a nucleus. There are receptors on that nucleus. You can bind hormones and other things and signals into that nucleus, and the DNA forms RNA, makes messenger RNA. Those messenger RNA will then code for individual amino acids and things, and we then stick all of those together, and that gets us a protein. So what we are hoping to do is to, by what we do during exercise, activate the right signals to make messenger RNA that will make actin and myosin, the scaffolding proteins, extracellular matrix, all of those things. And then get those messenger RNA turned into protein that will hopefully then get integrated into our existing myofibrils, and then we will have a bigger muscle. Okay? That's the process we're trying. So what we need to do then is we need to activate this transcription. And then we got to make sure that we have enough amino acids available so that when we drive transcription, we can actually translate those things into the proteins that we want. So there's a couple of things that we're going to do. There's also this idea of protein levels. Okay? Muscle protein, actin and myosin, myofibrils are always in a constant state of flux. I think I've told you guys, my dad was a high school football coach. One of his favorite quotes in the entire world is from a very famous football coach. His name was Lou Holtz. He coached in the NFL. He coached at Arkansas. He coached at Notre Dame. Lou's favorite saying was, "You're never as, Lou was, you're never as good as, things are never as good as it seems or as bad as it seems. But also, you're either going to get better today, or you're going to get worse today. You're never staying the same." Okay, that's the idea behind sort of hypertrophy and activity. Brady's muscles are either getting bigger or smaller today because they're in a state of flux. Okay, Brady, did you work out today? No. Did you work out yesterday?
I can't really work out.
Can't work out. Have you had some protein today?
Oh,
Okay. Brady may be in atrophy phase right now. Okay, but what is happening in your muscles right now is the net balance of signals that you have given it to make new proteins or to break down existing proteins. And so what we need to do with all of this is use hormones, diet, loading, stretch, metabolic stress, nervous system activity, all of these things that we can control with exercise and diet and supplements, et cetera, to try to tip the scales so that increasing protein synthesis predominates over protein degradation. We want anabolism to predominate over catabolism. That leads to hypertrophy. Okay? When these things aren't in the way that we want, then catabolism may predominate over anabolism, and then we get atrophy, and our muscles will get small. Okay? There's lots of things in your muscles that other tissues in the body would like to have. Muscle costs your body a lot of energy. So if you're not doing something with it, it may think, and you haven't eaten any protein, it may think, well, I will just scavenge some protein out of those myofibrils in my TA so that I can, make glucose in the liver or something. You've not had anything for a while. So this is where we are. This is what happens today and even tomorrow and even this week are not that important. Okay? It's always fluxing one way, minute to minute, hour to hour. What matters is over the long term, over a period of weeks and weeks and weeks, am I mostly anabolic or am I mostly catabolic? right? So I need to be anabolic from kind of the net of all of this for weeks on end to actually see hypertrophy. Weirdly, atrophy can happen really very fast. We can break stuff down way faster than we can put it together. We don't really talk about that very much in this class. If you want to know more about that, take spaceflight next. Take spaceflight next fall. Okay. So I borrowed this. I like kind of some of the things that are going to be in here. Okay. And so what this shows is all of the things that we think are going to contribute to some of the signals that drive both transcription of messenger RNA and translation of those messenger RNA into proteins. Okay, so we're not actually going to go transcription into protein. That's not how this works. It really should be transcription and then assign over here translation of this. But the signals may affect transcription itself. The signals may affect the translation of those messenger RNA. There is some interaction. But there are things that we know are driven by the mechanical load. Okay, how much weight are you lifting? How many times are you lifting? Okay, this is something, here we have what's called mTOR. All of the things that drive hypertrophy, this is the cellular hub or the signaling hub that drives hypertrophy. It's mTOR. We'll talk about mTOR. Probably not today in a lot of depth. We'll probably do next week. Okay, and then things associated with mTOR, P70S6 kinase. These things seem to be driven by load. There may be things we know if we get some hormones, there are nuclear receptors for those hormones, there's signaling, there's cell surface receptors for those hormones, we upregulate the receptors and things, we get autocrine and paracrine signaling from IGF-1, we get some endocrine signaling from insulin, testosterone, and growth hormone. All of these things may play some role in the midst of all of this. They are certainly, in a lot of instances, we just heard something called permissive. The hormone is permissive, like you need to have it in order to let the thing happen. If you don't have it, the reaction won't go. We're not sure that giving you more makes it even better, but it's the thing you've got to have it as part of the thing. And there seems to be some evidence that these things are at the very least permissive to allow some of this to happen. There are some things about neuronal activity. Calcium seems to matter. Calcium being bound to calmodulin and calcineurin, much like what it drives the adaptations in the nervous system, much like it drives sort of the breakdown of glycogen and those things. That stuff seems to matter. And then there may be some things that are activated by metabolic disturbances. Okay, AMP kinase, PGC1 alpha, stuff we've already talked about. Okay, these things and this sort of balance of metabolic disturbance with mechanical load that may either drive mitochondrial synthesis or drive hypertrophy. Okay, so there's a lot of things that are going to go into. So here's, in some ways, an expansion of what you saw right there. You guys don't have to know all of the things that are on. I just want you to see this and understand. What I've done is in the red square here is I put in place, this is mTOR. Okay. We seem to think that we have receptors on the cell surface that respond to mechanical loading. So disturbances that are going to be there from potentially high force levels or stretching or something. They're going to signal down, they're going to stimulate mTOR. mTOR signals through P70S6 kinase, and that may drive some initiation of things that are happening as we make the biopsies. Okay, but you can see there's also some things over here. There's IGF-1, especially that seems to drive differentiation of satellite cells, which also seems to be important. You need the combination of some of these things. Here's Wnt signaling that might also drive satellite cell differentiation. We have all of this stuff. We know insulin and IGF also signal through the insulin receptor, which may then also activate mTOR and all of these things. We know that you need leucine, which is one of the essential amino acid, I think. But leucine seems to be one of the more important amino acids that also interacts and helps to turn on mTOR. There's a lot of stuff that goes on. And there's a thing over here called myostatin. Myostatin is the anti-growth hormone. You get a lot of myostatin, and it basically shuts off everything, limits your growth. I'll show you guys some pictures later on. There are animals and people that do not make myostatin, and they're just jacked. It's called a Belgian blue bull. You guys can Google that if you want. There's myostatin deficient dogs and mice like people, some Olympians, very famously, they don't make myostat, and they just have these giant muscles, they're just ripped. Okay, and so there's some kind of integration of all of those things. Okay. So as we try to work our way through the things that are controlling all of these cellular signals that lead to transcription and translation, I want to break us into, we're going to kind of go sort of model by model through what we know through these individual ways of studying this. So we're going to start off by talking about what are called myogenic cell cultures. So this is growing muscle cells in a petri dish. Okay? A lot of what we know about how and why cells can become muscle, so the turning, the kind of initiation of making things into myotubes and then myofibers, comes from these models. Okay? We often start off our research on the cellular level. If it won't work in a myogenic cell culture, it's probably not going to work in a lower animal. It's probably not going to work in people. But there are lots of things that work here that don't go there. An advantage of myogenic cell cultures is that we can make all of our cells genetically identical. These are all type 1 muscle fibers from this particular genetic line. Okay, well, these are all type 2X fibers from this line. So we can make them all genetically similar. So that gives us a level of experimental control. Okay, it's easy in cell culture to control both the external and internal environment. You can bathe those cells in a certain amount of calcium or sodium, right? You can put it into a certain metabolic environment. You can control the amount of amino acids that it's given or what hormone it's given or whatever else. It's much, much easier in this environment to have tight control of those things, okay? But it's important to remember, this is not always representative of what we see, especially in intact human muscle. One of the things that very often lacks in cell culture is there's no nervous system innervation. And we know that's really important. And we know it's really important. If I cut the nerves going to Christina's hand, then the muscles innervated by those nerves will immediately begin to shrink. And it's not just from lack of contraction, it's because there's something that is helpful sort of about that signal. Okay? So as I said, there's lots of things that will make cells grow in cell culture that don't work. Yeah.
Is there a reason why I feel like whenever I read about that, it's always like C2C cells being used for these kind of things?
I know nothing about the individual sort of I don't know. Heather might know more. She might be able to tell us. We never used cell culture lines when I was a PhD, when we did a lot of hypertrophy stuff when I was a PhD student. My guess is that they grow easily. There's already existing data, and the lines are kind of already out there. You can buy them at a reasonable rate would be my guess, but I don't know why those cells are not small. So, really, really good question. They're probably cells that came from someone that had cancer somewhere in their cells. Yes, there's a book. It's called The Immortal Henrietta Lack, I believe is the name of the book. It's this woman, like they had really aggressive cancer, and like her cell lines have been like we've had her cells for 100 years or something. And there's questions like, is this ethically okay? Like, all of our stuff is done through these kinds of cells that just kind of grow and grow and grow. So it's a really interesting. Pardon?
Like took them without her consent.
That's the thing, is they took, that's right. I believe her name was Henrietta Lack.
Yeah, but they like called them fuel.
Yeah. So it's wild. No, I think she's been dead for a long time.
Cells are nothing.
If you think about it, tons of her like cervical cells, it's way more than any other somatic cell.
Yeah, it's really crazy, right? So
My mom used those in her master's research, and she read the book later and had no idea.
I didn't know.
She was like, yeah, everyone was using them. No one knew where they came from.
So, you know, I mean, it's all, it's kind of crazy. And you can think about all the scientific discoveries that we've made by using those cells that have hopefully led to good, but also just took somebody's cells without asking them. Was that okay? Right? Anyway, so there's a lot of kind of ethical concerns. But so just to show a little bit of these things, I don't know if anybody works with single cell lines at all or does any kinds of things. We have these embryonic progenitor cells in a lot of instances. And they tend to be PAX3 and PAX7 positive. And then we can move them into these satellite cells. We've got a quiescent satellite cell that's no longer PAX3 positive, it's PAX7. And then we can commit them basically to try to make these satellite cells that grow into, so I should back up. Most of these things are coming from satellite cells. Right? So the cell cultures come from these muscle stem cells, which progenitor cells. I should have probably said that. So you've got embryonic ones, they become satellites. A lot of what we have, we're getting them in this phase here. We can then commit them. When they become committed, they're going to express MYF5. They're going to then form a single sort of progenitor cell called a myoblast. It expresses certain things. Those things can then form into what are called myocytes. And then the myocytes will begin to divide and form what's called a myotube. And then once you've got a myotube, then the myotubes can either go together or get integrated into kind of existing sorts of things. And so you can look at these things that they're going to express. MyoD, myogenic. You can see when they get to be a myotube, they've got desmin, they've got alpha actinides, they're beginning to have some scaffolding proteins that are going to be there. We can grow them out from all. Okay. So this is what this looks like. These are just some kind of pictures down here of the various stages of what these things may or may look like. And as we go along here, then you can give these cells certain things or deprive them of certain things, and then from that, figure out, is this contributing or not? So, I mean, it would take, we could spend like 2 years just going through all of the work that's been done in sort of cell culture on things that may or may not cause hypertrophy. So we're just going to talk about the model a little bit and come back to a few of the things that we know. So once we see that things work in cell culture, most of the time, they're going to go into animal models, so models with rats or mice. So a lot of our cellular and molecular data is going to come from models like this. Okay, so it's good. They're genetically similar. Okay, we can very tightly control their activity and their behavior. We can tightly control their feeding habits. Okay, and if we wanted to, we can just cut out the whole muscle and study the whole muscle, figure out what's happening, not just in one fiber, but the whole thing. Okay. Things that are, I'm going to call them less good. Okay. Not that we have a bunch of rats and mice that we're going to have to kill after we've studied them for a while. But the methods for inducing mechanical failure are not always, I mean, you can make these things lift weights, but like you can't like tell a mouse to like go do bench press, right? You have to trick it into doing bench press or something. You got to put it on a wheel and make it run, but the wheel has extra resistance on it. Or you've got to make it like hit a thing that has it's got to press against it a certain force, right? And then it gets to eat or something, okay? And then, you know, every week you make it, it has to press harder if it wants to get to. So there are some things that we're going to do. That's the nice way of inducing it. Some less nice ones that we'll talk about, right? So it may not be necessarily ecologically valid at the same time. So one of the original ways that they do things in these, and we do some pretty terrible things to animals in the name of science, which probably needs to be done, but also always makes me feel a little queasy. We do a thing called chronic stretch. What we do with chronic stretch is we take Josiah's calf, we cut him open, we grab the medial head of his gastroc, and we basically stretch it and we fix it like farther up here. We move the sort of origin of it in a different place. So you cut it and then you stretch it and you attach it back and you turn loose again. Okay? Chronic stretch, you fix it at this long muscle. It's not what we do, right? We don't do this. No matter how much yoga you do, like, we're never gonna like fix your muscle just like stretched out like that. Okay? We find when we do chronic stretch, you get huge. These muscles get really, really big. So we think that there must be something magic about stretch. Now, Brady's cast probably isn't big enough to do this, but we do know in human models, if you do casting as a way to reduce activity and things, that whatever muscle gets casted at a lengthened state. So if you put yourself in a cast, I'll try to get myself in here, you put yourself in a cast like this, my bicep is stretched, my tricep is shortened. Put me in a cast like this for a couple of months. My tricep is going to shrink a bunch. My bicep's not going to shrink very much because it's stretched. You do the opposite, the bicep shrinks, the tricep stays the same. So there's something magical about stretching, right? That's going to play a role. But this chronic stretch model, we see some weird things. We see hyperplasia. We see some other kind of weird things from doing all of this. Again, it works, we get bigger muscles, but that's not what we do in people. Synergist inactivation, they just cut the synergist muscle, cut it out, or they'll denervate it. Okay, so we're back with Josiah's other leg, and in this one, we're just going to cut out his medial gastroc, or we're just going to see what happens to his soleus and his lateral gastroc. Okay, just remove one of your muscles. Okay, see what happens. So the same load then has to be borne over a smaller amount of muscle or a smaller number of muscles. So you're going to stress them more. Voila, they get a lot bigger. Okay. So we can do resistance exercise. When it's nice, it's voluntary. When it's not so nice, we knock them out and we electrically stimulate the nerves that lead to their muscles. We make them do exercise with E-step. And we can load them really, really, really heavily by doing all of that. Okay. So, because it's hard, I can't like get my daughter to do the things that I ask her to. How am I going to get a rat to do that work? So these are all ways that we can look at stuff that affects them. I mean, you could change how active they are. Otherwise, you can change what the hormonal situation is. You can change what the food situation is. But again, this is not exactly indicative of what happens in people. Okay, and then on to us, right? When we do mechanical or compensatory hypertrophy models where we're going to work out, you're going to lift weights very often, okay? You make yourself do more work, either by doing a lot of sets or very high weight or some combination of both. We get this, we know it works, okay? And so sometimes I feel this way. Other days I change my mind. So sometimes, like, we know that if you go lift weights, most of us are going to get bigger muscles. We know if you do some basic protocol, you're all going to get 15% bigger. So why do we care? Why do we care if it's 3 sets versus 4? Why do we care if it's 8 reps versus 10 reps? Why do we care about all of these other kinds of little things? And there are days that I think that probably is a good way to approach things. But the other days, it's, well, if we know exactly which of the things are the most important, then maybe we can optimize our dose for our athletes, or for our older individuals, or for our people rehabbing from a stroke, or something like that. Okay, maybe it's really important for us to know. These people can't do this, but this other stuff also works, so let's have them do it that way. So I kind of go back. Things that people have tested: intensity, right? So, like, what percentage of your max strength are you lifting? Does this matter? Probably. Volume. You can do 1 set, you can do 3 sets, you can do 1 day a week, you can do 3 days a week. What are you going to do? More here seems to be better. More here to a point probably also seems to be better. How many days per week? There's probably more to a point is also better. What kind of contraction? Well, we have some evidence that I'll show you guys that maybe eccentric contractions are important and help get more hypertrophy. But I defy all of you to go work out without doing eccentric contractions, or to go work out and just do eccentric only contractions, right? Nobody does only concentric stuff. Most people, you lift it up, lift it back down. Lift it up, you put it back down, right? And so again, we can experimentally manipulate things, figure some stuff out, but it may not always be that sort of applicable to what people do in their everyday life. And then there's some fascinating stuff about metabolic stress. We know that there does seem to be evidence that reaching a particular metabolic endpoint seems to drive hypertrophy, oftentimes of a similar magnitude, regardless of how much load you lift, maybe regardless of volume and frequency in these things. So this is an idea of what's called blood flow restriction. We'll talk about that. Mike's lab did a bunch of it, okay? Basically, you put a blood pressure cuff on, you restrict blood flow to the muscle, and you work out with a really light weight, but you get to fatigue pretty quickly. You do a couple of sets like that, and we see very often similar increases in muscle size to lifting much heavier weights. It's really fascinating. Really, really fascinating. Okay, so where to start? So what I'm going to show you guys is by no means comprehensive. What I'm going to try to do is hit a few of the high points. I'm going to show you a couple of kind of seminal studies. And I'm going to show you a fair amount of data from what are called meta-analyses. Okay? And a meta-analysis is a fancy review article where you essentially treat every study and the participants in it as their own participants. So I'm going to have 10 studies, and I treat each study like it's a participant in a bigger study. And so this study found a 10% change. This other study found a 12% change. You weight things and then you kind of average out across all of these studies. Here's what we found. I'm going to show you guys some stuff from some metas that may hopefully also guide you a little bit on what works. Okay. If you're going to go work out today or for the next 8 weeks, how many days a week should you go? How many sets should you be doing? How much weight should you be lifting as a percentage of max when you do all? Okay. So here's a really interesting study from, it was published in 2002. It was performed at the University of Ohio. Okay? Not Ohio State. This is a University of Ohio by a group of guys, a group of faculty that were buddies with my PhD advisor. We had a girl that was a master's student that came to our lab and actually participated in this study. But it was one of the first ones that I can remember, that I can find, where they took muscle biopsies, but they had people train in 4 groups. They had a group that did low repetitions, so high percentage of max strength, low reps. Okay, 3 to 5 reps, 4 sets. Okay, lift heavy. They did some intermittent stuff, kind of 9 to 11 or 12 reps for 3 sets. And they did high reps, low weight, very low weight, high reps for a couple of sets. They're trying to kind of even out the amount of volume that these people are getting. And they had a control group. And they looked at the effects of this on overall changes in size, okay? But then also in fibers that were type 1, type 2A, and they called them 2B, but they're really 2Xs, okay? So this is 8 weeks of training. They did squats, leg press, knee extensions. So they're really hitting the knee extensors. 2 days a week for the first month, and then 3 days a week for the second. So what I want you guys to look at, what I think is really, really interesting, is here you have control, low rep, intermediate, high rep, and you've got pre and post. This shows changes in the fiber type expression. So look at this. In our control group, no real changes, but in all 3 of our groups that train, your 2B fibers get shifted over to 2A fibers. Okay, we've talked about this before. This is the thing that happens. This is resistance training that drives away 2X fibers. Okay. Again, this had been observed, but it had not necessarily been observed using all of these different rep ranges. Okay. Heavy lifting drives that conversion. Light lifting, what some people might think about as like just doing metabolic training, also drives those changes. Here we have been, these are cross-sectional areas, average cross-sectional areas of the type 1, the 2As, and the 2X fibers. Okay. And so what I want you guys to see is that if we look at these things, and they really should have done these as a percent change, but what I want you all to see is that low reps, high weight, and then our intermediate reps and intermediate weight caused hypertrophy in all 3 fiber types. Okay? All 3 fiber types. Then we look in the high rep group, They're getting a little bit bigger, but it's not enough to be statistically significant. So these were some data, and we have some newer-ish data. I said again out of Stu Phillips' lab. Although they may not be exactly comparable, that potentially high rep, lighter weight doesn't cause hypertrophy. And it doesn't really matter that much between the low rep, high weight, and the intermediate reps. So some of the first evidence showing what happens, and it also shows that in general, all three of our fiber types are all hypertrophy. Okay? Now, if you look at this, you may think, well, Dr. Black, the type 1s aren't getting as big as the type 2s, and that may very well be the case. But you use your type 1s all the time. And so they may already be at a bigger, sort of a higher percentage of their max at rest, kind of from normal daily activity than the 2As and the 2Xs. Okay? So some really kind of interesting and cool data, I think, on trying to tease out the effects on the individual fiber types of these different. Okay. So here are some things. And I just really like these graphs. Okay. This is not necessarily the most inclusive review articles, but what we see here is these are, each data point represents a single study. And what we have, this is the quads. These are the biceps or the elbow flexors. And what you can see on the x-axis of each graph is the intensity, the percentage of 1 rep max that was used. And then the y-axis, in an effort to try to normalize across all of these studies, they took it and they figured out what was the increase in cross-sectional area per day of the length of the studies. Some of these studies were 8 weeks, some were 12 weeks, some were 16 weeks, some were 10 weeks. And so their ability to try to make comparisons, they just figured out how much bigger are you getting per the number of days that you're actually exercising. Okay? So here we have in this what looks like a relatively positive linear relationship between the intensity at which you're working out and the magnitude of increase in muscle size. Okay? And if you squint really hard in the elbow flexors, maybe you're seeing the same that's kind of going on here. All right? But especially in the quads, when people are going between, say, 50 and about 100% of 1 rep max, which very much falls within the normal daily recommendations that we see from the American College of Sports Medicine or the National Conditioning Association, higher weight does seem to be a little bit better. Okay? Higher weight does seem to be a little bit better. Okay. But they're all getting us some level of hypertrophy. These three that are outlined way down here at the super low intensity, I don't really know. These things are weird. I think one of these is a blood flow restriction. It's a control group from a blood flow restriction. The other one is the basic campus that I just showed you guys. And so these are a little bit of bets are off down here. Okay. Elbow flexors, everybody's kind of grouped in the same area. We've got these people working out at 108%, which makes no sense to me. What they were doing, but again, if you squint hard enough, maybe there's a little bit of a relationship. That's cool. Okay. So if we think about intensity, there is some evidence that maybe working at 70 or 80 or 90% of your maximal strength is a little bit better than working at 50%. Well, that's still doing fine. It should still work. Okay. So this slide now is going to look at volume. Okay, volume. So this is the amount of weight you're lifting as a percentage of your max multiplied by the number of repetitions that you're doing in a given week training sessions. Okay. So here are people that did concentric, eccentric. So this is traditional dynamic exercise, traditional weightlifting. And here are people that trained on like an isokinetic dynamometer, so like KinCom or a Biodex or something. Okay, so if you look at this one though, those of you that have had stats, does this look like there's much of a relationship here? Jordan and Drew are shaking their heads no, right? Shotgun blast. What's on here? I don't know. Pretty random, right? The one that's got the biggest has one of the kind of lower end numbers, some folks there. So we look at all of this, and it gives us some evidence that maybe volume isn't as important as the amount of weight that we're lifting. Okay, so doing a whole lot more isn't always better than doing a little bit less. Okay? At least if you look across all of this. But again, this is difficult to try to quantify because we don't really know for sure, right? It's hard to match up what people are doing in all of these different studies. Plus, these data are more than 10 years old, and so they don't always capture some of the stuff that we've done. Now we're going to ignore the journal in which this was published and focus on the actual data. So these were published in the Journal of Pure Power, which I've looked up. It's a real thing. It was theoretically peer reviewed, but this is an actual meta-analysis. So they calculated the effect size. I have no idea why it was published there, why it wasn't published in like the Sportsmed or some other place. This one provides us a little bit of contradictory evidence on volume. This is looking at studies that did one set of exercise. How much hypertrophy they got. So you're going to come in, you're going to do one set of 8 reps today, okay, versus doing 2 sets or 3 sets of it. So here we look, doing multiple sets in a given session gets us a larger effect size of change. Okay, they're both still moderate effect size, but a little bit more of an effect than just simply doing 1 set. And then if you break the multiple set one down into 2 to 3 sets versus 4 to 6 sets, there seems to be this dose response that we're going to see there, that maybe doing 4 to 6 sets is a little bit better on top of doing the 2 to 3 sets. But again, there aren't that many studies that are in this bucket here, so it's a little bit confounding. I choose to look at these things not as does the number of sets and volume matter. I choose to look at it. Look what you can get from just doing 1 set, right? You got to be efficient, people. If you're going to have kids, wait till you have a kid. You got like 10 minutes. All of your daily things that you get to yourself have to occur in that 10-minute period. Do I want to read a book? Do I want to exercise? Do I want to take a nap? You got like 10 minutes to yourself. You get quite a bit out of 1 set. Okay? So things to keep in mind. So additional things to put on. Okay. This is exercise frequency. So the number of days per week. So again, we're splicing out this idea of volume and stuff a little bit different. So I don't know what the hell these people are doing. 12 sets of 12 sessions per week. So like you're going to go 6 days a week and go twice. You're going to go 4 days a week and go 3 times on each day. I don't know what those people are doing. But if you throw this one out, boy, there's not a lot, right? But again, everybody does 2 days a week or 3 days a week or 4 days. Like everybody's kind of all clustered together. So it's difficult from the Wormbaum's paper to kind of make, are there any relationships? Maybe if you think about it, the bicep one's kind of nodding that direction. The quads, maybe not so much. But here's some really cool data, and we will save this for next week, because this will take me like 5 minutes to explain. So I don't want to drag us into all of that. Okay, so let's stop here. All right, we will shut this down. You guys can